Welcome to today's PNOC Foundation webinar to provide information from the PNOC Atrt Working Group about the Atrt Registry and why this is so critically important for progress with this disease. This webinar will share updates from the last webinar we hosted in November of last year. I'm Bruce Campbell, and together with my wife Alan, we co-founded PNOC Foundation 10 years ago, 3 years after our own son George was diagnosed with a pediatric brain tumor. Our vision is to lose no child to brain cancer and to improve the quality of life for kids and their families fighting and surviving pediatric brain cancer. PNOC Foundation is unique in that it's the only 501c3 exclusively dedicated to supporting our partner organization, the Pacific Pediatric Neuro-Oncology Consortium, otherwise known as PNOC. The now global network of scientists and clinicians successfully collaborating and translating the latest biological findings into innovative clinical studies and trials. In 2019, PNOC established unique disease-specific working groups with the main intent to bring clinicians, scientists, philanthropy, and families together to enhance clinical trial development. The Atrt Working Group brings together the world's leading physicians and scientists who have expertise in Atrt and are dedicated to improving the outcomes of children with these tumor types. They feel very strongly that the best way to understand a rare disease such as Atrt is to gather as much data as possible and have that data evaluated by a collection of expert physicians and scientists. Today, you'll hear from PNOC's two leading principal investigators about the Atrt Registry. But first, we want to acknowledge all of the patient families and Atrt advocates joining us today. Many of you have been involved in the incredible fundraising effort that has occurred since we launched the dedicated campaign for the Atrt Registry in March of last year. This group has inspired their communities and creatively raised funds and awareness at local sporting events, partnering with a national jewelry chain, and even a tie-in with NASCAR. Thank you and well done. We at the Foundation are here to support any current and future fundraising ideas you have for the Atrt Registry. For more information, you can reach out to us directly or email us at info@pnocfoundation.org. Today, we also have the great honor of hearing from a few Atrt patient families. To kick things off, it's my pleasure to introduce two of the world's most esteemed Atrt experts. Dr. Ashley Margol is a pediatric neuro-oncologist who specializes in the care of infants, children, and young adults with brain and spinal cord tumors. She holds a master's degree in clinical biomedical and translational research from the Keck School of Medicine and has continued her career in clinical and translational pediatric brain tumor research. She's an attending physician in the neuro-oncology program and division of hematology oncology and blood and marrow transplantation at Children's Hospital Los Angeles, and an assistant professor of pediatrics at the Keck School of Medicine at the University of Southern California. Dr. Margol is the principal investigator of the PNOC Atrt Research Group. Dr. Alyssa Reddy is a pediatric neuro-oncologist with more than 20 years of experience in diagnosing and caring for children with central nervous system tumors. She has specific clinical and research interest in neurofibromatosis and embryonal tumors in very young children. As a PNOC member, Dr. Reddy is involved in the development and conduct of cutting-edge clinical trials with the goal of improving long-term survival and quality of life for patients. She believes these goals are best accomplished through a collaborative approach from a diverse team of scientists and healthcare providers with the ability to offer new therapies through clinical trials. She is the co-principal investigator of the PNOC Atrt Research Group. Welcome, Dr. Reddy.
Thank you so much, Bruce. And thank you everyone for joining us today. We are really excited to update everyone on the activities of the Atrt Working Group. I did not share the right screen, so sorry. Let's do that again. Okay, we have the right screen now, full screen. Great. Again, thank you everyone for joining. I know it's morning for some, afternoon, and even evening for others, and we really appreciate you making the time to, in your day to join us. So my part is going to be relatively brief. I know some of you have already heard this before, but for those who haven't, I want to go into a little bit of just brief detail about the working group itself. So we established the working group several years ago, and we set some very clear goals in terms of ATRT. Our goals are listed here. We want to obtain a better understanding of factors that affect outcome. We know we have been able to improve survival for patients with ATRT over the last few decades, but we know that it's really not good enough, and that a lot of children, unfortunately, don't survive their disease. So we really need to understand what treatments work and for which patients, and that's one of our major goals. Along those lines, we want to identify the best therapies for patients. We want to increase awareness and funding. We know that this is a fairly rare disease, but when you break it down and you realize that it's actually the most common malignant tumor in children less than a year, it's brain tumor, that is, it's not really that rare. And so it's really important with rare diseases to increase awareness. And of course, we want to support our families and our patients with ATRT and do everything we can to not only cure them of their tumor, but learn and also give them the best possible quality of life. So as this group came together, we developed sort of two main programs. The first one is our Atrt registry, which we've been working on for the last few years. And Dr. Margol will go into a lot more detail about where we are with that. But we're very excited about the registry because we believe it's going to serve as a foundation for studies that we do in the future. We are also, while we're developing and launching the registry, we're working on an early phase trial. We want to be able to move early phase trials quickly through the pipeline. So we're developing a phase 1 and phase 2 trials specifically for patients with ATRT. This is our working group. It includes investigators who are both basic science investigators working on laboratory discoveries, and clinical researchers, really for people not only in the US, but around the country. And this is a list of the people who are part of the working group. But in addition to this group, there have been people from all over the world, really, who have come to our meetings, who shared their research, who shared their ideas. And this has truly been a real collaborative effort and will continue to be so. We know from the families, from the community, from all of the stakeholders, from the patients to the families to the researchers, that we really need more data. I've been in this space for quite a while now, for 20 years, and we know that we're learning more about the science, and even within ATRT, there are different subgroups, and the science and the technology has really improved. But we still don't know which unique variables about a patient's tumor or about the patient themselves, which really predict how the patient is going to do, and in order to be able to figure that out, we need lots of data. We need to try to put together data from as many patients as we possibly can to understand this in sort of a timely fashion. We know that patients can't wait, and we can't continue to spend decades to really learn small amounts of data. So we need to really put data together that's very detailed so that we can better understand these tumors. So these are the things that we are working on and know need to be done. And we also need to identify new therapies that hold promise, but we need to see how they specifically affect ATRT. We've talked a lot previously, and I think most people know this, but just very briefly, some background about ATRT. About 75 children are diagnosed in the US each year, and that almost all of these patients are less than 3 years of age. We have learned, and several groups have shown, as I mentioned, that there are 3 molecular subgroups. This is based on methylation profiling. And we know, for example, in other disease types, such as medulloblastoma, we're now stratifying treatment and risk based on subtypes. We haven't quite gotten there for ATRT, And again, we think this is a data issue. The more data we can put together, the better we can understand about these subgroups. As I mentioned earlier, we have made some progress. This graph here in the left lower panel, this is a survival curve from the Children's Oncology Group ACNS study, ACNS 0333. As many of you know, this was an intensive protocol that included high-dose chemotherapy and radiation therapy. And what we were able to do in this study was we were able to increase survival compared to historical controls. We know that now roughly half of the children who are diagnosed with ATRT and treated with an intensive treatment regimen will survive. But that's not good enough. Half is better, but it's still certainly not good enough. And none of us are willing to stop there. We need to learn more. And we also know, as you can see from this graph, is that many of the patients who relapse, the jagged line is actually the solid line is what we call event-free survival, which is usually a reflection of relapse. That most of the patients who relapse very early. So again, we need to do more research to really understand who these patients are. There have been several studies that have looked prospectively at treating ATRT specifically. These are them listed here. You may be familiar with one or all of these. I mentioned ACNS 0333, which was the Children's Oncology Group study. There's also a regimen and a study that Dana-Farber has done. And then there were 2 studies through St. Jude. Just to show you from this summary, is that these studies somewhat yielded different results in looking for prognostic indicators. So for example, on the Dana-Farber study, patients who'd had a gross total resection did better, as did those with posterior fossa tumors. But actually that was not borne out in the other studies. Similarly, in ACNS 0333, our patients with sonic hedgehog tumors actually did very well and had no relapses in the first 6 months. But again, this was not borne out in other studies. So at this point, we really don't know why that is. Does that have something to do with the treatment, that differences in each, or does it have something to do, or do we just not have enough data to really be able to say something concretely? And right now, we believe the later is really the issue. So here are the current issues, just quickly. Patients with ATRT have only been really studied individually in a handful of prospective clinical trials. I just mentioned the 4 major ones. We have yet to clearly identify prognostic clinical variables, and survival remains poor, with, as you saw, only about half of patients having long-term survival. The data is conflicting. And again, the current studies that have been done and the data sets really just are not large enough to answer the questions. And we really need to be able to combine these treatments to figure this out. ATR is relatively rare, and so that adds challenges to sort of what we will call the traditional clinical trial design. And we believe that we want patients, our goal is survival and cure. And so we really, randomized trials are difficult in this space because we really don't want patients to get less therapy at this point in time. We need to establish, and I know Dr. Margot is going to talk more about this, but we need to establish an external cohort of patients with ATRT. This is really the best way that we can push the field forward and increase survival for more patients, cure more patients, and ultimately, as Bruce said, our goal is to have no child die from a brain tumor. So with that, I will turn it back over to Bruce.
Thanks, Dr. Reddy. I do want to share with the audience that we will actually have a Q&A at the end of the session, and there is a Q&A button, I believe, in the chat. Just bear in mind that this isn't a forum to necessarily answer or give individual consultation about cases. Certainly scientific general questions we can answer, and if there's a desire to set up a consult for a second opinion, then that's possible to do after the webinar as well. But I do want to turn to and introduce the first of our patient families. Natalie is the mother of Vincent from Pennsylvania. And I believe that Vincent was diagnosed with ATRT 5 years ago at around the age of 4 months old. So it's just such an honor to have you on, Natalie.
Thanks, Bruce. I appreciate having the opportunity to share Vincent's story with other parents in particular. So Vincent was 4 months old when he was diagnosed in June of 2018, actually. It was just 5 years ago last week. He started off with a very slight head tilt, and he was misdiagnosed initially as torticollis. We live 15 minutes away from Children's Hospital Philadelphia, so it was never a question to us on where we were going to go for treatment. It was just automatically we were going to CHOP. Everybody, all these parents, we all go through this. It's devastating. And so I'm going to try to cover Vincent's story and answer most of the questions that I get from other parents about his treatment and what exactly we've kind of chased. So when Vincent was 3 months old, he had the head tilt. We thought it was torticollis. We took him to physical therapy. At his 4-month well check, his head circumference had grown significantly. So our pediatrician sent us right to the CHOP emergency room. That was his only symptom. He wasn't vomiting or anything else. He just had a very slight head tilt. When we were there, we actually found out he had 2 brain tumors. One was a 9-centimeter brain tumor in the right parietal temporal occipital lobe. He was 4 months old, so it was 9 centimeters. The other one was smaller at the posterior fossa. He immediately had a debulking surgery on the 9-centimeter tumor. They removed about 40% of it. And then he started the modified IRS-3 or Dana-Farber protocol. He did respond really well to the chemo, and about 6 months into that, at the end of that front line, he had, the tumors had shrunk and he had grown. So we did 2 more craniotomies, 2 separate, a week apart, and achieved gross total resections on both. Vincent was declared no evidence of disease. We continued on that protocol, which had about another year of what they call maintenance on IRS, but not maintenance on what we think about today. And about 4 months into that, he suffered a rare reaction to one of the maintenance drugs, and he went into acute liver failure. It was VOD type symptoms, and he luckily did recover from that, but that meant that he couldn't continue on that protocol. So while we were consulting with many other institutions, our doctors and ourselves, we had him on oral Tamidar to bridge him for about 4 months. Then we decided to do 1 cycle of the ACNS high dose stem cell rescue. So he had done that. He was about 14 months old at that time, completed that. He did really well through that, and we ended treatment. He was about 20 months old when we actually ended his treatment, and he was, he had a lot of side effects, but everything was very manageable, and he was doing well and thriving. But unfortunately, 5 months later on his surveillance scan, we found another tumor in a 3rd location. It was in the pineal region. We did another craniotomy. That was his 4th craniotomy, got a gross total resection, and immediately started a MAMAT type protocol that we had been recommended to us from sick kids. He started that, tolerated it really well, did great. 5 months later on a surveillance scan, there was a 4th tumor in a 4th location. We did another gross total resection, 5th craniotomy. We immediately started tazemetostat commercially, off trial, not using it compassionately in the pill form the day after his surgery. We coupled that with intrathecal topotecan monthly intrathecal chemo through a lumbar puncture because he has a shunt and not an Ommaya. We did that for about 18 months and he responded beautifully. He was doing well, thriving, had very little side effects except some thrombocytopenia in the beginning with the platelets. At about 18 months on the treatment, he went in for a surveillance scan, and in the background of his spinal imaging, they found a very large mass, a very large mediastinal mass. He had no symptoms. His blood work was fine. there was a lot of confusion as to whether or not this was a secondary cancer or something else going on. Eventually, we did biopsy it. It was determined that it was a T-cell lymphoblastic lymphoma, believed to be a result of treatment-related tazemetostat and or in combination from all of the treatment he's had over the past, at the time, 4 years. That was right before he had turned 4. Our neuro-oncology team immediately stopped the tazemetostat and brought in the liquid oncology team at CHOP. They kind of had to regroup together and creatively come up with some type of a treatment protocol, which we started immediately. Vincent went into remission very quickly from the lymphoma, and he has now been in remission for about a year from the lymphoma. He has about a year left for that treatment. We couple his lymphoma treatment continued with the intrathecal topotecan and another drug called bortezomib. And he's been doing very, very well. His ATRT has now been stable for 3 years as of July of 2020. It'll be 3 years since his last recurrence. He has never had a recurrence in any of the same tumor beds. It's all been different locations. He does have the germline mutation. His tumor subtype is TYR. He has never had any radiation at all, ever. And he does have a shunt, like I said, and all of his intrathecal chemo has been through lumbar puncture. He just turned 5 in February. He's making incredible gains in his development. He's walking independently now, and we just registered him for kindergarten last week. So I was, like most of us, without hope 5 years ago. I would have never believed we would be here registering for kindergarten today, but we don't know why he responded the way that he did, but he has, and I'm grateful for all of the research that everybody on this group, working group is doing. And that's why I feel like this, ATRT registry is so important, because of all of this information. I have parents from all over who contact me to find out what I did to get here. And I don't have an answer, but this is what happened. And it needs to be shared so that database can be there for the world to look at, and we can really try to make a difference in finding cures for our kids.
Natalie, thank you so much for sharing that incredible story. I mean, like all of our kids, Vincent sounds like an amazing fighter, and I think your story highlights, so much about pediatric brain cancer and this ATRT disease, and so thrilled to hear that he's celebrating the milestone of enrolling in kindergarten. That is really amazing. I want to take the opportunity to switch now to Dr. Ashley Margol, who's going to share with us an update on the registry protocol.
All right, let me see if I can share my screen appropriately. So thank you all again for the opportunity to give you guys an update, kind of where we are on our ATRT registry. And I think the stage has been set beautifully here with this example of Vincent and his case, and how, ATRT therapy works for some kids, different therapies work for other children, and we're still kind of trying to figure all of this out. And so you heard Dr. Reddy say the word data over and over again, kind of in the introduction, and that's kind of really the crux. And when we sat down as a group and thought about designing a trial many years ago, we realized, we need to take a step back, and really we need to collect data. So you're going to hear the word data throughout the rest of this hour. So the point of the ATRT registry is to collect data. And what data are we collecting? I don't want to be trite and say everything, but that's really the goal, is to collect everything. So we want to know the comprehensive clinical data. That means how the patients presented, all of the different therapies that they received, the toxicities that they had from therapy, et cetera, et cetera. We are going to collect imaging, and not just imaging reports and measurements, but the actual MRI images. And we're going to house those so that we can go back and look at them ourselves. Because we know that things change with regards to criteria for what we call progression or response, and we want to be able to go back to the source. And then we've talked about collecting tissue and molecular information. We know that molecular subtype is of great interest, and so we really want to be able to at least have that information. And for patients who have tumor tissue available, we would obviously be very interested in collecting that for additional research in the future. So kind of once we create this, kind of the question is, what are we going to do with it? And so there's two kind of big categories of ideas. So the first one, as you heard Dr. Reddy mention, and we still really don't understand kind of what variables give a patient a better or a worse chance of responding to a particular therapy, right? So there's so many variables, whether it's how much of the tumor came out, where the patient had metastatic disease, or if they had metastatic disease, we talked about molecular subtype. And what about a combination of these, right? So what if you have a patient with sonic hedgehog subtype that has a gross total resection and no metastatic disease? Does that mean that that patient is a good risk factor patient or not? The truth is we're still very, very much in the dark about this. And a lot of it is just due to patient numbers. So you'll hear us say, we need more data, we need more data, but that's really the crux of it. So the goal is that with collecting as many of these cases as possible, we're really going to be able to answer hopefully some, if not all, of these questions. And then you also heard Dr. Reddy mention this idea of external control cohorts, and I will do my best to explain this as simply as possible in the upcoming slide. But the goal really is to be able to come up with novel clinical trial designs for patients with ATRT, for the reason that, especially in the relapse setting, we don't have effective therapy. And also when you think about trying to design a randomized clinical trial for when there's only 75 patients every year in the United States, you can imagine how long something like that is going to take. And we really just want to be able to be more efficient so that we can get these therapies to children in better and faster manners. Okay, so you're going to have to bear with me a little bit while I kind of walk you through this idea. So several people have said, well, how does this registry differ from other data sets or data sources? And there's this idea of something called patient level data. And that means that for each particular patient, we can link the clinical data, we can link the tumor tissue, we can link the MRI images, rather than kind of all this data being put into a pot and then making kind of aggregate assumptions. And what I mean by that is sometimes in a clinical trial, we get a bunch of information in, but we can't get patient level data out. So can you tell me what happened to patient A? Can you walk me through their timeline? What about patient B? Can you walk me through their timeline? And it's kind of a product of just how the clinical trials were designed, what the purpose is, and really a big issue is just resources to try to collect that level of data. But if we're talking about being able to design an external control cohort, patient level data is really, really the key. And if we can harness this patient level data and we can collect it, that's going to lead to improved therapeutic trials and ultimately to improved outcomes. So again, here's just an example of kind of patient level data, right? So you've got your patient, you've got their family medical history, you've got their tumor tissue along with the molecular information, you have their actual imaging, and you have their detailed treatment information. And then ultimately you have their survival data, but not only survival data, you have quality of life data. And I'm going to highlight this just for a minute because, hopefully at some point we get to a place where we've got pretty good cure, pretty good therapy, and we might start asking the question, okay, these two therapies have similar cure rates. Is one therapy less toxic than the other? And that's where we really want to be capturing this quality of life data so that one day we might be able to recommend one therapy over another because actually has a better quality of life or toxicity profile. So we feel like that's important. And to be honest, a lot of this kind of quality of life survival data, neurocognitive data doesn't exist for the ATRT cohort currently. So again, this does not exist right now. And a lot of people have said, why? We have all this data. And you heard Natalie say, like, she has all this data about Vincent and people have been coming to her. And this is a story that we hear over and over. The truth is it seems like it should be a simple solution, but unfortunately it's not. It really requires tremendous amount of resources to collect this type of data. And it's not only to collect it, It's also to standardize it. So when we say date of diagnosis, that has to mean the same thing across the board. Is that the date of the MRI? Is that the date of the surgery? We also have to be able to have this data in formats so we can easily share it, right? This data should be shared. That's the whole goal. So we want to be able to have our data set talk to other data sets, and that turns out incredibly complicated. So I'm going to give you kind of an example, and I shared this before, I think on the last webinar or in other forums with some of you here, kind of what does it mean to actually use an external control cohort. So when you think about a randomized trial, you enroll a patient, and that patient gets randomized to, let's say, the standard of care therapy, which we'll call arm A, or the treatment therapy, which we'll call arm B. And a lot of times that's a one-to-one randomization, meaning the same number of patients get randomized to arm A as they do to arm B. Well, the idea here is that instead of having this whole group of patients that are randomized to arm A, you could simply pull those patients out of a database. So if we use ACNS 0333 as an example, and we say, okay, we want to compare this brand new therapy to the regimen that was given to patients on ACNS 0333. And let's say we're just going to do it for gross totally resected M0 sonic hedgehog patients. So we would design our study, figure out our sample size, how many patients need to be assigned to the treatment arm, how many to the control arm, and we would find those patients who meet this criteria. So gross totally resected sonic hedgehog, non-metastatic, patients with ATRT that were treated, on ACNS 0333, and we would pull those out of the database, and that would serve as a control cohort, so that all the patients being enrolled on the study could actually get the therapy. And this is a pretty novel design. It's being talked about more and more in a number of different settings, including in a lot of conversations with the FDA that Mike and I and others have had. And so we're really trying to kind of harness this idea when we think about clinical trials moving forward, especially in the relapse setting where we know that we don't have effective therapy. So kind of this study design, this is complicated. It's not really meant to be, it's meant to be comprehensive, but can you see my cursor if I point?
Mm-hmm.
Okay, great. So the goal is, again, is to collect as many patients as possible. And so kind of this cohort here is going to be patients previously treated. And these are going to be patients who survived or succumbed to their disease. We really want everybody to be able to participate, and we understand that all data is precious data. There's going to be a cohort of patients who are newly diagnosed or currently undergoing therapy, and then a cohort of patients who are relapsed. And the only reason that these are separated are that patients who have relapsed disease are going to have this option here for an expert tumor board. And that's going to be a group of us from the PNOC Atrt working group, as well as the patient's treating physician, if they choose to attend. And we'll really just review the imaging, the clinical course to date, as well as the pathology molecular findings, and give some guidance about potential therapy options. So for all patients, regardless of cohort, again, we'll be collecting all of these things here. So there'll be full clinical data collection, MRI for central review. So we'll have a neuroradiologist who's looking at all of those MRIs. For patients who are getting lab draws as part of their standard of care therapy, we'll get some blood from them specifically to look for germline SMARCB1 mutations or alterations. If patients are undergoing CSF collection for clinical care, then we'll be collecting some of that CSF. We'll collect tumor tissue for molecular subgroup classification. This is a requirement for enrollment on this study unless this was already done and the data are available. So we feel like really knowing this molecular subgroup is key. And then I mentioned quality of life assessments. And again, kind of this whole data set, especially a lot of the clinical data collection forms, were created with input from our colleagues at the FDA. So we've had a meeting with them where we show them our data collection forms and asked for input. And they've been very helpful in trying to help guide us on that. And then again, data sharing is really important. So there's a number of national platforms through the NCI, which we'll be collaborating with to make sure that these data are available for additional researchers. So who can enroll? Any child who had or has been diagnosed with ATRT. And as I mentioned, we must have tumor tissue available or results of molecular subgroup determination. And we can do that centrally. Again, if it hasn't been done, we'll do it most likely through the NCI. So how will patients enroll? If the patient or child is treated at a PNOC institution, then that PNOC institution will be able to directly enroll that child. For subjects, patients treated at non-PNOC institutions, we're going to have a central enrollment site at CHLA. Jasmine Polly, who's on today, and here's her photo, so that you recognize her. Feel free to reach out to her. We have an email account set up for you to send us inquiries, and we're going to be kind of asking that you help us gather a lot of this information. So we'll need tumor tissue or a pathology report, the actual images on a CD, and then we'll be requesting full medical records. And Jasmine's really going to be key with working with a lot of the parents and local treating physicians to make sure that we have all of the information that we need to enter into the registry. And then I'll just wrap up with a little bit of the timeline. So we are really, really excited that the registry is moving forward. We plan to open to enrollment, at least initially, at UCSF and CHLA by September 1, and then we'll be opening at other sites shortly after. I'm gonna stop.
Fantastic. Thank you for sharing that update. It's time to hear from our next patient family, Kate from California, who is the mum of Frankie, who we've known for several years now. I'm really happy to report. You know, I saw a picture of Frankie recently and didn't recognize him. He's got so big. Welcome, Kate. It's great to have you back on with us.
Thank you. Yeah, I'm Frankie's mom, and he was diagnosed about 7 years ago. So it's been a while, and we've sort of been through the roller coaster of all of this. And as I've been listening to all this data stuff, I've been thinking a lot about what it was like to go through his disease in the beginning and how, you know, he passed with flying colors. He didn't have any regrowth, and he had a total gross resection, and it went really well, as you know, as medically from a medical perspective. But it would have been really great to know that he was going to do really well. I don't know if that will ever come about, and if there will ever be peace of mind if your child gets diagnosed with brain cancer. But part of me is thinking like that would have been really great to know that here we are 7 years later, our nerves are completely fried and he's doing great. But I just really appreciate what's happening with PNOC and how these groups of just brilliant minds are getting together and, you know, tackling this disease that's so rare. Frankie's doing really well. He's 10 now. I have this picture. I wonder if you can see. He's doing so good, and he's huge. And, you know, he has a couple side effects. He is, you know, he had some growth issues, but now he's like back on track, and he has some, I guess it's epilepsy. He hasn't had a seizure in a year, but we deal with some side effects, but overall, like, we are very happy with the outcome of what we had to do. I guess I just want to say that this ongoing research, I just appreciate it so much. And I had the honor of going down to, I can't remember what the event was right now in my mind, but I came down and I saw all these doctors and researchers and support team all together. And I just really appreciate that. And I really hope that future families can benefit from what's happening.
I think that was the scientific symposium we had.
Yes,
yesterday.
yes.
Yes.
And I got there towards the end when everybody got to relax, but I was able to see everybody's smiling. I won't go into detail. I got to see some of, the presentations that were going on, and I was blown away by just how detailed everything is and how everybody's putting so much effort into this rare disease that, well, I guess it's not really, in my world, it's not rare, but yeah. Just want to say thank you.
Thanks for sharing, Kate. Yeah, for those of you who may be joining us for the first time, PNOC Scientific Consortium hosted a global medical symposium here in San Francisco in May and brought together over 100 of the world's leading pediatric neuro-oncologists. A part of the work that they do, and it's one of the things that is just so incredibly unique about this organization, is the level of global collaboration that's happening to find answers to these rare diseases. And it only happens through, philanthropy, unfortunately, and the continued support of audiences like this one. Dr. Margot, was there anything else to add about the trial in federal funding?
Yeah, Bruce, and I have just a couple slides because I know we had talked about this before, so I wanted to give people an update. So I'll share my screen again. But again, I know many of you have participated in conversations about this before, and you know that kind of one of the big challenges in trying to fund something like this is it's not that sexy, right? So it's not some like fancy immunotherapy study where there's lots of different grant opportunities, and so pretty early on we realized we were going to have to get creative about how we were going to get this funded. And so we're really lucky to have Susan Wild's office, who, has been a really big supporter of pediatric cancer funding initiatives in the past. And so we reached out to her and some other members of Congress and said, hey, we're trying to figure out a way to fund this. Can you help? And her office put this letter together, and I've showed this before, but basically the point is, we had a number of members, both Democrats and Republicans signed on to this letter, which really encouraged additional funding through the Childhood Cancer Data Initiative to specifically go to this Atrt registry. And I think this is like a huge win, and I'm really proud of this. And I think it really shows the commitment of so many people, pretty important people, recognizing that this is a really important cause. And from this, we actually got some language into the omnibus, and I just put it here for you to see. It's a little bit not exactly the way that we wanted it to go, which isn't that surprising. It goes to markup with the Appropriations Committee. And basically, the way that this was ultimately worded was that this money was kind of to continue to support enhanced efforts through the CCDI to focus on ultra-rare tumor types, such as ATRT. So there's a little bit of wiggle room here and wasn't quite as direct as we had wanted, but we are kind of still in conversations with the CCDI to see how we can leverage this language to direct more funds towards our efforts. That being said, we're not resting on our laurels, and while those conversations continue, we are obviously still trying to explore as many different options as possible. And not unlike most trials in PNOC, the funding for a study such as this really is going to be multifaceted, right? We're going to have to pull from multiple sources. And so just to talk a little bit about the federal funding right now. So we're still having conversations with CCDI. Mike, Adam Resnick, and I actually went back and met with a congresswoman last week when we were in DC, and she is incredibly committed to this cause. We've been in contact with her healthcare legislative assistant. We're going to go back and see her in July, and she is really willing to kind of go above and beyond to figure out how to help us with funding. It's going to take a while. I don't know exactly what it's going to look like, if it's going to be specific to ATRT or rare pediatric brain tumors, but I think it's really important for the community to know that you do have advocates who have hopefully a little bit of power. And then we are working on additional grant funding. So there's a cancer center supplement, which we submitted. due tomorrow. I think it's already been submitted. We're applying for DoD funds and some other things like the Children's Cancer Research Fund. So we're continuing to do as much work as we can. And then obviously we're incredibly grateful. This group has really kind of blown us away with the amount of philanthropic support that everyone here has provided. So again, so incredibly appreciative that you think that what we're doing is worthwhile and that you're continuing to help us fundraise for the efforts.
Oh, that's a great segue to our next patient family, who in their short time of being part of this community have made an incredible impact philanthropically. So I'd like to introduce Erica from Colorado, whose teenage son, Finn, was diagnosed with ATRT I think in January this year.
Thanks, Bruce. And thank you for giving us the opportunity to participate and just for all the work that this community is doing. We are very new to this journey, and it's incredibly helpful to hear the stories that Natalie and Kate shared. And I look forward to giving you the 5-year and the 7-year and the 10-year update. Our son Finn was diagnosed in January. He skied Vail on January 1st with his friends, and 13 days later was having a biopsy that came back with results of the ATRT. And since then, it's been a pretty wild 6 months. We feel very lucky and grateful to be in proximity of Children's Hospital Colorado, in some care of some great pediatric oncologists there. But we have tried a lot of things. So he has had a couple surgeries, not gross total resection, but partial resection, a few weeks of focal and CSI radiation, I think 7 weeks total. We've tried ACNS 0333. We've also tried the St. Jude protocol, and he most recently completed a bridge of ice because those protocols were not showing the effect that we would like to see. So he actually wanted to join and express his gratitude for the work that's going on and the continued focus on this. Unfortunately, he was readmitted to the hospital last night for a fever, but I just wanted to share that we are all very grateful, and we are immersing ourselves in learning and the community and exploring options for not only a very rare disease, as has been shared, but I feel like Finn might be the only person in the United States right now who's 18 years old and has this. So a shout out to anyone out there who also has teenage children who have experienced this. We would love to connect with you. So thank you to all that PNOC is doing.
Oh, thank you, and thank you for sharing the story. And we're obviously, rooting and praying for Finn, and hope that he continues to do well. And as a resource, we're here to help you and your family any way we can. I'd like to actually introduce PNOC Foundation's Director of Development, Adam Eversole, who's going to talk to us a little bit about continued funding for the Atrt Registry.
Thanks for the introduction, Bruce. So I'm going to talk to you guys a little bit today about the where we are as far as funding goes and where we need to go for the projects that Dr. Marble and Dr. Reddy has talked about. First and foremost, though, I just want to thank all of the donors, the fundraisers, the patient families who have made this possible. We have moved quicker on this than we thought we were going to be able to because of your donations. So thank you all. We can't say it enough. We would not be launching this registry this summer if it wasn't for your support. So thank you. When we look at the fundraising goals for the registry, so our initial goal to launch was $200,000, which we have surpassed, which is why we are launching this summer, like we just heard. In 2024 and 2025, we're going to have a larger goal of $125,000 annually. And that That is going to be basically to make up for previously diagnosed patients that we now need to enroll in the registry, as well as enrolling newly diagnosed patients that will be diagnosed in 2024, 2025. Once we get through that, the goal is going to be, we think, about $75,000 a year. And that's going to allow us to enroll 80% of newly diagnosed patients on an ongoing basis. When you break this down, again, over here, we've got our initial goal, $200,000. We've done that. And then 24 and 25 of that $125,000 annually, we have a commitment from Team Beans each of those years for $75,000, which is incredible. We're so grateful for them and their support, leaving us with $50,000 each year that this community and the PNOC Foundation will raise. So I'm very confident in our ability to do that. We can't do it without you. We won't say thanks enough, but $50,000 for those 2 years, and then approximately $75,000 annually moving forward. And this is just another slide that breaks down that total when you take into account the initial money needed to launch, which we've done, and then the 2 years that we're going to do catch up and enroll previously diagnosed patients. That total is $450,000, and we've raised $350,000 of it. So leaving that $50,000 a year equaling the $100,000 gap, which we're in a good place. We're really excited. What comes next? So, you know, as Dr. Margol pointed out, the registry is just the beginning, right? We need the registry to allow us to move on to better, quicker, and more successful clinical trials that are going to eventually cure this disease. And to do that, we're going to need a lot of funding. And individual philanthropy, institutional funding, grants, federal funding, corporate philanthropy, foundations, all of this is going to be critical to create really a mosaic of funding that is going to allow us to take this to the next level. So this is certainly not on you guys. You're a small group that have gotten us really far, but we want you to see this and know that while we still need your help, we're looking at every single avenue possible to make all of these trials a reality. How can you help? You know, like I just said, you already are. The peer-to-peer fundraising that this group has taken on is why we are launching this registry this summer. You know, when you remove some of the very large top commitments, you know, the average donation was under $200 a donation. So an incredible example of what community and grassroots support can do. So what can you do? Continue to peer-to-peer fundraise, host an event in your community. No event is too small. Golf tournaments, bowling nights, bake sales, it all adds up. Share PNOC's information on social media, via email with your friends. Connect PNOC to a corporate sponsor or a foundation that supports a work like this. You know, I am always happy to get on a Zoom or get on an airplane and talk to someone about the work that we're doing and explain to them why, if you want to cure pediatric brain cancer, PNOC is the absolute best investment that you can make. So please connect us to those folks, and we'll tell them what we're doing. And the last thing, of course, is donate. You know, most people on this call already have, and your donation means so much to us. And please continue to make PNOC a philanthropic priority for you and your family. And lastly, we really just want to close again by thanking all of you. We're so grateful for this group of people that have gotten us this far. You know, there are too many individuals to name, but Team Beans, Team Finn, Hope for Atrt, Noah's Legacy Foundation, the Wood family and community, Kinsley's Unicorn Squad, Team Vincent, Team Jocelyn, and Casey Masiantonio, and more. You know, there are anyone who set up a fundraising page and contributed to this cause. Thank you. We're so grateful. The amounts were incredible, and we're just thankful to have such a community of people that are supporting this really important work. So I'll close there. We can't thank you enough, and everyone on this page and everyone not on this page. We're so thankful. You know, my information is on the screen, so please, if you ever have an idea that you want to bounce off of someone, if you have a fundraiser that you're planning and you need some support from our end, we're here for you. So please email me anytime. I would love to have that conversation. I'm really grateful that I got to speak with you all today. So thank you, and I hope to hear from you soon.
Thanks, Adam. I think, Dr. Margol, we're going to go back to you to talk a little bit about the interventional study.
Yeah, I think, Adam, thanks so much for making the point that really, the goal of the registry is to answer all these questions, but the ultimate goal here is to really, improve therapies, and that's where this needs to go. And so I thought I might take this opportunity then to kind of pivot just a little bit away from kind of talking about the registry, and show you how the registry is really going to integrate into an interventional study. So I'm really excited to show you guys this slide because this is new information for anybody who's on, and just got approved to proceed through. It was approved by the PNOC PIs, and so it's going to come to fruition as a protocol. So we are designing an adaptive clinical trial for patients with relapse disease. And what that means is that we're going to have multiple arms of the study open. And we'll have as many arms as we have good therapies. And so we'll enroll patients who have relapsed ATRT, and they will go on any one of these arms, and kind of the arm that they go on will be decided by a variety of factors, kind of depending what the therapy is and what the patient has already had. And if we ever get to a point where we can stratify by molecular subtype, then we will certainly do that. So we're kind of designing this whole study, but right now we have our first arm. And I have two friends here. So this is Eric Robb and this is Jeff Rubens, and they're both researchers at Hopkins. And this is really based on a lot of their data with paxilosib, along with gemcitabine data that really came from some of our colleagues at Children's Hospital Colorado, and also across the ocean in Princess Maxima. And so we're really excited that this is going to be the first arm of the study. More details to come, but it's going to be a combination study of paxilosib and gemcitabine, and it's going to be a phase 2. So we already have safety data for both of these drugs in children, and so we're not expecting a lot of overlapping toxicities with the combination, and we're super excited to have this kind of heading into protocol development in the next couple of weeks. Tom Rosenberg from Dana-Farber is going to be leading this arm in particular, and Susan Chi, who many of you all know, is going to be helping kind of with the overall study as well. And just to give you a quick idea kind of what that looks like from a study timeline, I have a quick timeline here for you. So we're going to start with protocol development here. I'm hoping starting next week, and hopefully getting approval sometime between winter and spring of 2024. And our goal is to really have the study open next year. Obviously, we'll be discussing funding support for this study as well, but we're really excited to kind of have an interventional study on the horizon. So I wanted to be sure that we were able to give you guys that update hot off the press.
That's awesome. Thank you for that. I know we're coming up to the top of the hour. We only have 1 or 2 questions in the chat, and I think to help us fill the questions today, we are very happy to have PNOC Scientific co-founder and Professor Emeritus, Dr. Mike Pradas, with us.
Yeah, thanks. I know, Alyssa, you're typing, but maybe it would be good for the whole group to kind of listen to you to respond to this, because this is a very important question that families have. If a child comes in, the question is, if they come in and they have a gross total resection and then start high dose induction chemotherapy, but the tumor then comes back, the question is, would you still continue with that same strategy after another resection, or would you move on to some other therapy at that point?
Yeah, I think it's hard to, answer like individual questions, like to do a real justice and provide what we feel is good. So
But generally speaking.
I think, again, it really depends on the age of the child and what therapies the patient has had, what we might recommend, and again, at what point, were there any delays in getting to a specific point? So I'm a little hesitant. Actually, I don't know how you feel, but I'm a little hesitant to answer that question without seeing more information about the case that I feel like perhaps the family is referring to.
But this then actually emphasizes one aspect of the registry is the optional molecular tumor board, right? Because that would be an environment or place where specific case histories could then be discussed by a group of folks like you that could give individual recommendations. And I want to emphasize, we didn't really talk about the molecular tumor board too much, but that's going to be a very important, I hope, resource for families for very unique, specific questions like that.
Absolutely.
And yeah.
And I think it will give us the chance to, like, we'll have the actual images in front of us and we'll have the whole history, which is really what we need to make those determinations. So we're excited to be able to offer that tumor board as a good way for us to kind of help answer a lot of these questions, because we know there's so many of you that have questions like this out there. So we're hoping that you come to us in that forum with them so we can be of assistance.
The follow-up question is actually critical to this entire discussion. How close are we to finding subtype specific therapies for ATRT? I mean, if you could imagine in the very best case scenario, let's say we get a good external cohort, and that's going to take time with good patient level data that's specific enough that everybody agrees it's comprehensive enough. And then you have this platform study. Maybe just a general sense of how long do you think it might take, knowing the small number of cases there are.
You want to guess, Dr. Ray?
Yeah. Just how soon, I think we could sort of, I think we all have sort of our biases based on, our own experiences, what we think might work. And some of the work, like, for example, the gemcitabine work, suggests that some subtypes may be more responsive, but they all seem to respond. So again, because we, especially in the relapse setting, we don't have, really good therapies that provide prolonged response or, stability. So I think we're probably at least a few years, honestly, from being able to do that. Obviously, that would be a goal if there's clearly a signal. But when you get people that work in, but work more on the sort of basic translational science, and when you get to what the SMART-B1 gene, the gene that's defective in these tumors, what it actually does, there's sort of a debate about whether, how to best approach that. And is that something that the type specific should get the different types should get a different therapy for, or should we really try to target that, because we know that what that defect does in terms of something called chromatin remodeling.
I think I can just add very quickly, Bruce, before is that we have to also become, as a community, smarter about our trial design. And there are ways that we can minimize the number of children on a specific therapy, on a specific arm, to say whether it's a go or a no-go, right? Could be a futility question. It could be a success question. And there are statistical ways of trying to manage that. So it doesn't take us 10 years to get to an answer. Maybe we can get there a lot sooner with a lot fewer children enrolled. The other component that PNOC is really trying to champion, and we are championing, is global outreach, because if we're just dealing with only children in the United States, we're missing out on hundreds and hundreds of children in other countries that can inform just as well about the therapies. And we're working with partners in many countries to try and optimize that speed chart, if you want to think about it, so the timelines are much shorter.
So yeah, Dr. Prados, thank you very much for that. We're over time. I know that our doctor panelists literally have patients that they need to treat, so we've got to let them go. I want to thank all of our panelists for joining today, for their contributions. A particular shout out to the patient families that were able to join us and share their stories. To everyone who joined us today, we hope that you found today's webinar informative and hopeful that there is progress happening in real time. This is an amazing group of people that are dedicated to improving outcomes in this area. If we didn't get to your question, we will actually answer it after the webinar ends. And if there's anything we can do for you, any further information, feel free to reach out to us at info@PNOC Foundation. Thanks so much to everybody again for joining, and we look forward to talking to you soon. Thanks.